are platelets. Okay? Depending on what growth factors or what chemicals our body is secreting, the stem cell can turn into any one of these. Okay? Have you ever heard leukemias? Right? Leukemias are blood cancer, basically cancer of any one of these cells. Okay.
So stem cell can turn into any one of those?
Yes, depending on what chemical our bodies release. There's specific chemicals for each pathway it goes to. Okay? So a stem cell is said to be a precursor cell. Okay? This is why there's a lot of research going on stem cells. Because even as an embryo, you had stem cells, and those stem cells turned into maybe heart tissue, brain tissue. Does that make sense? Okay? So this is why there's a lot of research on stem cell, because you could cure a lot of diseases. Now, let's talk about white blood cells more in detail. So we're done talking about red blood cells, now we'll talk about white blood cells. Now let me ask you this. Just a name, granulocytes or agranulocytes. So white blood cells are split up into two main categories, either granulocytes or agranulocytes. What do you think the difference between granulocyte and agranulocyte is, just by the name? Exactly. So granulocytes mean those cells within the cytoplasm, they contain granules. Agranulocytes mean those cells in the cytoplasm, they don't contain any granules. So right here, you can see these are cells, these are granules. Okay? These are little stuff that actually, when they release, they're chemicals. So we have three types of granulocytes: neutrophil, eosinophil, and basophil. Neutrophil, I mean, there are multiple characteristics, but the two I want you to know. Okay, hold on. Neutrophil, when you actually look at it under microscope, okay, it has, when you're looking at the pH, it has a neutral pH. This is why it's called a neutrophil. Okay? Neutral. A basophil, or eosinophil, basically has acidic pH. Well, can't say it has it means that acidic dye tends to color it. And the basophil, basic. You can kind of, it has a basic pH. It tends to work or get coralized while basic dye. And when you go in microbiology, you learn about different type of dyes. We have acidic dye, basic dye, neutral dyes. This is why they have a different color. Okay? Basophil tends to look more bluish, okay? Where acidic ones tend to have more of like a purple or pink color. Okay, neutrophil. First, to combat infection. So anytime you have an infection, it's the first cell to arrive at the site. Neutrophil is the first one that comes at the site. And it tends to protect you against bacteria. It's basically armed to protect you against bacteria. Now, a neutrophil has basically, if you look at the nucleus is multi-lobe. That's just important, multi-lobe. A eosinophil, its lobe looks like sunglasses. It has two lobes, bi-lobed. Okay, it looks like a sunglass. It's an easygoing day and you have your sunglasses on. You know, you're on a beach, it's an easygoing day. That's how I always remember which one is which. Basophil, you can't really see the nucleus because there's so many granules that it tends to cover up the nucleus. Like when you look at it, you can't see the nucleus. It's very hard to find the nucleus because it has increased accumulation of granules. So, first one to combat infection, it loves to fight or protect you against bacteria. It has a neutral color and at a neutral pH, and it's multilobed. Eosinophil has more acidic, it looks more pinkish purplish. Eosinophil has, it's a bilobed nucleus, the sunglasses, I remember. It protects you against parasites. So anytime you have infection by a parasite, it tends to protect you. It's also responsible for allergic reactions. So eosinophil is also responsible for allergic reactions. Basophil. Basically, as I said, it's very hard to see the nucleus because it's compacted with granules. Basically, it has more of a bluish color, and it's more basic. Again, protects you against parasites. Okay. It's very hard to find a basophil in the bloodstream. Okay, it's a rare cell. Rare cell. It's also responsible for allergic reactions. So, protects you against parasites, responsible for allergic reactions. It's a very rare cell. It's very hard to find the cell when you look at the bloodstream. Specific thing about its granules. Its granules contain heparin. Okay, now let's talk about agranulocytes. Agranulocytes are cells, their cytoplasm contains no granules. Now, there are different types, but the first I'm going to talk about is just macrophage. Okay? So agranulocytes, you have two main classes. You have monocytes and macrophage, and then you have lymphocytes. Okay? There are two main classes. So this slide just focuses on monocytes and macrophages. Monocyte, macrophage, like here. Now, what is the difference between monocyte and a macrophage? Monocyte is basically a white blood cell which is found within the bloodstream. As soon as that monocyte leaves the blood vessel, enters a tissue, it's no longer called a monocyte, it's called a macrophage. Okay? So, within the bloodstream, it's monocyte. As soon as it leaves the bloodstream, enters the tissue, is called a macrophage. So monocyte, bloodstream, macrophage, tissue. Okay. Macrophage is called a phagocyte. Have you ever heard about phagocytosis? Okay. Phagocytosis is a process in which a white blood cell will engulf a microbe and destroy it. It's like a Pac-Man going through and eating it up. Okay? So a phagocyte is any cell that is able to perform phagocytosis. Does that make sense? Phagocytosis is a process where a white blood cell will engulf a microbe and destroy it. So it will eat up a bacteria and destroy it. It's like a Pac-Man going through. Okay? So phagocyte is any cell which is able to perform phagocytosis.
On number 2, right next to it, what is that? Macrophage. Oh, tissue. Tissues.
Now, lymphocytes. Now there are 3 different types of lymphocytes. I only located 2. The third one is NK cells, which are natural killer cells. Okay? So there are 3 types of lymphocytes. T lymphocytes, B lymphocytes, and natural killer cells. I'm not getting in detail about natural killer cells. I'm just going to focus on T and B. That's what I'm about to put that in. Now, where does T and B lymphocytes produced in bone marrow? Produced bone marrow, or red bone marrow to be more precise. Okay, so T lymphocytes and B lymphocytes, both of them are produced within the red bone marrow. but they tend to mature in a different area. B lymphocytes tends to mature within the bone marrow, red bone marrow. So where it got produced, that's where it matures. But a T lymphocyte matures in the thymus. It matures within the thymus. So one goes to a local school, another one sends off to a boarding school. Okay? Function. Now, anytime I was thinking how do I want to go right now? Function of T lymphocyte is cell-mediated immunity. It's responsible for the cell-mediated branch of immunity. Function of the B cells is humoral response, or humoral branch. Now we have different types of lymphocytes. We have TH and a TC. TH stands for T helper, and TC stands for cytotoxic. So there are two types of T lymphocytes. There's a TH, which is helper T cells, and then there is cytotoxic, which is TC. Okay? Now, anytime you hear cell-mediated, imagine a cell-mediated is where the cell itself is doing the killing. Okay? So it's like an army personnel actually getting into combat directly. Anytime you hear about humoral, it deals with the production of antibodies. Yes.
When you say cell-mediated,
It means that the cell itself is like in combat with the microbe and doing the killing. With humoral, that's whichever cell belongs to the humoral group, they don't actually go and fight the microbe. They actually tend to release something and then destroy it. So it's like throwing a grenade. Okay, the grenade actually kills it. That person is not actually in a physical combat, whereas T cell is actually in a physical combat. This is why they call cell-mediated, where the cell actually does the work. This is humoral because they actually release chemicals which destroy it. Which the chemical is antibodies. Now, a B cell will transform into a plasma cell. A B cell will transform into a plasma cell. Okay, so plasma cell from B cells. So it's a mature B cell. So a B cell will transform into a plasma cell, and only then can it produce antibodies. Produce and secrete antibodies. So if a B cell knows there's infection and it has to secrete antibodies, it first has to mature into a plasma cell. And only then will it be able to secrete, produce and secrete antibodies. Okay? Now, we have different types. So antibodies deals with the humoral response. Production, plasma cell. Types. There are 5 types. IgA, IgG, IgE, IgM, IgD. Okay? So there are 5 types: IgG, IgA, IgD, IgM, IgE. Now I'm not getting into the structure of antibodies. We'll cover that in microbiology. The function, I'm not even going to tell you what the function of each one is. We'll cover that in physio. What else is just a big, broad picture about antibodies. Its function, it has multiple functions. Either it activates the immune system, or it could neutralize toxins. Okay, neutralization of toxins, activation of the immune system. That's all I would say right now. Okay, each one has kind of a different function, but. So neutralization and activation of the immune system. I'm not making you responsible to know what is the function of T helper cells or T C cells, okay? I'm just trying to simplify it right now. It's already involved. Okay, now last thing we're going to talk about is blood groups. Now, who knows their blood group in here? A positive. O positive. B positive. AB negative. Okay. So what is a common thing you hear? No, I mean in a sense, sorry. When somebody's saying the blood type, you hear the letter, either A, B, or you hear the positive and negative, right? So when you talk about blood groups, there's two main systems. There's the ABO blood group, which talks about the letter, and then you have the RH blood group that deals with the positive and the negative. Okay? Both of the systems are combined together, and you tell your blood group. You know, you don't just say I'm B blood group or I'm negative blood group. You always say either B positive or B negative. You always combine the two. Okay? So first we're going to deal with the ABO blood system, and then we'll talk about the RH system. So now these are my red blood cells. Okay, all four of these are my red blood cells. Now this is my plasma. Now, if I say somebody has blood group A, okay, that means on their red blood cell, on the surface of the red blood cell, they have the A antigen. The antigens can be either proteins, or it could be lipids, or it could be a combination of lipid and proteins. Okay? Glycolipids, glycoprotein. So it's usually a carbohydrate with a protein, or it's a carbohydrate with a lipid. Okay? Those are what usually antigens are. Now, antigen is anything that stimulates the immune system. Okay? All of us are humans right here, right? But how do I know? Okay? So these two girls, get up. Oh, man. Okay? Okay, turn around. Now, how, they're both humans, they're both females. But how do you know this is Jennifer and this one Stacy? Okay, let's say you knew them. How do you know when you look at them that this is Stacy, not Jennifer, or vice versa? Come on, you guys. Okay, one is blonde, one is darker, what else? The height, right? The race, okay. So do you see those are identifying things? You guys can sit down. You see that's an identifying thing? You can look at a person and tell? You guys can sit down. Oh. Unless you guys like spin.
Oh, we're Oh!
They just like all that attention to them. Okay, so antigen is like something that it's personal to you. It kind of identifies you. Okay? Does that make sense? Yes.
What are the A's?
Those are antigens. Okay? So antigen, as I said, the composition could be it could be a glycoprotein or a glycolipid. A glycoprotein is a carbohydrate bound to a protein. Glycolipid is a carbohydrate bound to a lipid. Okay? So that's the composition, either a glycolipid or glycoprotein. And an antigen is supposed to identify you. So a person who has a blood group A, on the surface of the red blood cell, they have the A antigen. Okay? But in their plasma, they have anti-B antibodies. So antibody against the B antigen, that's what it means. Does that make sense? So a person who has blood group A have the A antigen on the red blood cell, but in the plasma, they have the anti-B antigen, anti-B antibodies, meaning antibodies against the B antigen. Because when these antibodies come in contact with that antigen, they will destroy it. So the anti-B antibody, when it comes in contact with the B antigen, it will destroy it. Does that make sense? Same concept. Let's say this is B, blood group B. What antigen is on the surface? B. What antibody do they have? Anti-A antibody. Okay? Good. Now let's say somebody is AB. What antigen do they have on their surface? What antibody do they have? No, because if they have an anti-A, it's going to look at this antigen and kill it off. If they have an anti-B, it's going to look at this antigen and kill off the red blood cell. So no antibody. So that person's plasma contained no antibody. A person who is blood group O, what antigen do they have on their surface? O? They have none. The O stands for 0. So a person who has blood group O have no antigen on their red blood cell. Okay? But what antibody do they have in the plasma? Both anti-A and anti-B. Anti-A and B antibodies. So they have both. Does that make sense? So from here, which one is universal acceptor? AB. AB. Acceptor means receive. So a person who is AB blood group, they're universal acceptor. Why? Because they don't have any antibodies. Even if I give them blood group A, there's nothing to kill off the A antigen. Even if I give them blood group B, there's nothing to kill off the B antigen. Which one's a universal donor? Oh, why? Because their surface does not have any antigens. So even if I give this blood group to this person, they don't have the B antigen, so it can't get killed. They don't have the A antigen, so it can't get killed. Does that make sense? When you transfuse blood, you don't transfuse the entire blood. You only transfuse red blood cells. You don't do the plasma. You don't do the white blood cells. You don't do the platelets. So you only do red blood cells. Okay, now RH blood group. So red blood cell. There's a plasma. So RH is an antigen. So a person who is RH positive, what antigen do they have on their red blood cell? Negative. They have the RH antigen. So the positive tells you that they are positive for that antigen. Positive means yes, I have it. Negative, no, I don't have it. Okay? So a person who is RH positive, meaning on their red blood cell, they have the RH antigen. What antibody do they have in the plasma? No RH antibody. They don't have any RH antibody. A person who is RH negative, what antigen do they have on the surface? They don't have no RH antigen. So they don't have the RH antigen. But what antibody do they have in the plasma?